Well, I'd like to thank Dana and Jennifer for setting up the workshop. So I don't know if you guys know how lucky you are for a workshop to have them guiding it, and because there's like decades of experience there, to have Dana, all his background in genome editing, and then to have Jennifer's background from the beginning of CRISPR, all the way up to now. So it's really great. So it's really, I appreciate you guys putting this on. I think the talk's supposed to be about 30 minutes. I know it's late, so I'll try to kind of move along. This is sort of just a visual map of what I'm going to talk about. So I'll talk a little bit about group 2 introns at the beginning, very briefly. Then I'll go over ZFNs, homologous recombination, and what we did with single-stranded donors. And then I'll spend most of the time on CRISPR and oligos, and then look at design, length, mods, and things that you can put on them. So I'll spend most of the time here. And I'm going to try to be brutally practical here. So in terms of you trying to come to the workshop and learn how to use it, things that you can go back and take back to the lab and try out. And then I'll end with just some brief comments on HR limits, so homologous recombination limits in certain cell types, and then some effects of aneuploidy. So at Sigma, in about 2004, we licensed this technology from Alan Lambowitz at the University of Texas, Austin. We called it Targetron. It's a group 2 intron. And so unlike a guide RNA, which is only 120 bases or so, this is sort of a monster. It's about 1 kb long, has lots of secondary structure. But the cool thing about it, like CRISPR, it's RNA guided, so it has a couple of loops. They're not contiguous like on a guide RNA, but it has a couple of loops that can be retargeted. And amazingly, it's amazing that Alan ever figured out how this worked. With all this, all this real estate for the RNA to look at, he zeroed down those 2 pieces to being the ones that interact with the genome and target this to insert into the genome and knock genes out in bacteria. So we've marketed that system. It's the first thing that we worked on at Sigma. So I mention it here because this is where we first got into genome editing at Sigma in 2004. So, and the cool thing about it is it's independent of homologous recombination. So this works in lots of different bacteria for knocking genes out. So the big reason we got it was to see if it would work in mammalian cells. So in 2004, 2005, there was no gene knockout technology for mammalian cells, and we tried to get it to work there. And we could get it to work a little bit, but never enough for practical genome editing to work. So unlike CRISPR, I think we've been somewhat amazingly lucky with how fast CRISPR was adapted in the mammalian cells. Because this was a bacterial system, CRISPR is a bacterial system, and it seems to have translated really well. So we shifted over to ZFNs very quickly after that. So after we couldn't get the system to work, we licensed ZFNs from Sangamo Biosciences. And I'm just showing this as one of the first collaborations we got involved with. There was a scientist in my group who's on this paper, Xiaoxia Sui. She started looking at RNA, and we collaborated with Sangamo and a few of their other external collaborators to make the first knockout rat. So that was a really exciting time. We've done a lot with ZFNs, but I'm not going to talk a whole lot more about that today. So what I really want to talk about is oligos, single-stranded oligos. And I'll just start off with talking about the simplicity and the inspiration with them. So when I was in graduate school and I first started working with DNA, whenever I would order an oligo, I would have to handwrite every base onto like a form. And then I would take that and fax it. So I'd feed it into a fax machine, right? And then like magic would happen. And then like a few days later, I'd get a FedEx envelope with all these oligos in it, and then proceed to try to build something out of it. And so when I first did that, I was fascinated with it. I was fascinated that I could specify what this was, and then send this electronically out into the ether and come back and then build something biologically. So it's amazingly simple. So that's really, once oligos were made, that whole technology, long time ago, there's been a lot of researchers that have been trying to get oligos to work, to combine them with human cells to make targeted mutations. So I can't go over the wealth of literature. For this slide, I really wanted to dig up a list of references, but it's just too much. There's a lot of chemical modifications, people have tried to put into oligos, and a lot of treatments to human cells to make these work together, but it never seemed to come together. So when we brought ZFNs via Sangamo into Sigma in 2007, we set out to distribute those to the research community. We started to learn about how to make donors. This is what donors looked like at the time we got ZFNs at Sigma. You'd have a plasmid that would have 800 base pairs of homology on one side, 800 on the other. And This would take several weeks to make, and you'd have to sequence verify it, and it was relatively expensive for every experiment you wanted to do. So we did this a few times just to get our feet wet with it, learn how it worked. But we're also wondering, will single-stranded oligos work as donors at ZFN cut sites? And we were almost very discouraged to try this because it had failed so much in all the previous research in the literature. And the first experiment we thought we would try to do with it, and this is one of the practical things I want to show you in terms of you going back to the lab if you've never done this. You can make an oligo that's got a left homology, right homology, and then some restriction site right in the middle. And so how you want to design that is so that this site, whatever site you choose to be here, will be right at the ZFN cut site or right at the CRISPR cut site. On the genome, wherever you want to cut. And so what should happen is the cut will happen and this will get integrated. And then what you can do is PCR the cells, the pool of cells, and then you can cut with that restriction enzyme and detect whether that was incorporated or not. So this is a very simple experiment. So we tried this with human cells. So I had Fu-Cheng Chen in my group try a variety of cell types with this. So this is the oligo-only sample. So this is the PCR product. If we were going to see any gene targeting happen at all, they should all show up down here. And so you can see in all these different cell types, there's nothing here that showed up at all. So, but when we added AAVS1 ZFN in, we got a great amount of activity across a lot of different cell types. And this was one of those moments where, I didn't really believe the data when I first saw this because so much had been done with oligos before. But I was just astounded at the frequencies that we saw here. And for full disclosure, we're not the first ones to think about using oligos and ZFNs at the time we did this. There were a couple of other papers where people had tried it. But the ZFN platform we had from Sangamo was really good. We had a lot of really highly active ZFNs that could cut at frequencies of 30 or 40%. And that's one of the reasons we tried this as well, because we thought maybe that would help somewhat. And in K562 cells, we got upwards of 50% targeting in those cases. So, and if you can see here, it's pretty variable across these different cell types, and I'll come back to that in a little bit. So so Fu-Cheng showed me these first examples, and I didn't believe it in the beginning. So what we did is we single cell cloned everything out to actually show that those HinD3 sites were actually in single cell clones and that you could actually modify them. So that really kind of made our brains go poof in terms of all the things that we could do, because to really start playing around with this, you just had to order other oligos, and it's very easy to think of something and then order it and redesign it. So we did a lot of variety of different experiments and then published them in Nature Methods in 2011. And I can't go through all the experiments we did here. There's only 2 primary figures in that paper, but there are 18 supplemental figures that we put into that paper. And there's a lot of good stuff in there that we couldn't put in. Like we did specificity, we had some extra sequencing data and stuff like that. So you can dig into that. One of the other primary figures besides what I just showed was we really just wanted to show how practical it was to make, say, a codon conversion at a kinase. So we had a collaborator that we met through a ZFN workshop, Morten Froden at the University of Copenhagen, and he was working on this RSK2 kinase. We had already made a plasmid donor for him, so we made this single-stranded oligo donor, and it seemed to work wonderfully as well, and that's all delineated in the paper. Okay, so that worked for ZFNs. So now I'll move on to CRISPR. So this is where I want to get into more of the practical stuff. I'm going to go through all these sort of frequently asked questions that we run into. So in terms of, do oligos work similarly for ZFNs and CRISPR? How long should they be? Should it be sense or antisense? How far from the cut site should the mutation be? And then I'll talk a little bit about chemical modifications. So first, in terms of CRISPRs and oligos, they work at really high efficiency. So this is just some raw data that was produced by Yiping Huang and Xingzhou Ji in my group recently, probably the last 4 or 5 months or so. And these are CRISPRs that are cutting at the AAVS1 locus, and we can see robust integration that's on scale with what we saw for ZFNs. So it works, and there are probably 30 plus independent publications that show that oligos and CRISPRs work as well. So there's a lot of validation out there. So we haven't done a lot of detailed direct comparisons of ZFNs and CRISPRs. And it's like a lot of the other methods you've seen. I don't think anybody wants to really tease apart those. They just want to see it work, at some high frequency. Okay, so how long should these oligos be? So Yiping and Xingzhou recently did this for CRISPR. So they took several different oligos where they had a CRISPR cut site here. They had a HND3 site going right into the cut site, and then they just varied the length of the homology on either side. And so what they found, what they recommended was that you can see cutting from anywhere from 40 to 200 base pairs of total homology on either side. So that would be 20 on this side, 20 on that side, or say 100 on either side. And they recommended that the ideal length is 70 to 160. And I guess it depends on the company that you order the oligos from. Once you get up above 120, sometimes you can run into problems in terms of quality of the oligos or how long it takes to get them. So I think for most applications, you should be able to stick around 130 or less. But that's it.
Any idea why efficiency goes down at longer lengths?
No, I don't. No, that's brand new data for us. We never went that high with ZFNs. I don't know. So I'm not sure. Yes?
Instead of making the tree, if we put the last side, like a 34 nucleotide, should we increase the homology arm?
I have heard of people putting LOX-P sites in with oligos. So I've heard of people trying to do that with mice.
But still, even if it's 30, instead of 30, 50 nucleotide, the homology arm can be 40 nucleotides?
I believe so. I don't know if exactly 40 is the case, but the upper limit, you're looking at pretty much the upper limit of oligosynthesis here. At the time I heard about the LOX-P work, I think the longest oligos I'd heard of were 130. So you're probably looking at 60 or 70, on either side of the LOX-P side at least. I don't know, no one's done a detailed study to know if it matters, plus or minus 20 base pairs on either side. But I've heard of it working with LOX-P, so. Okay, so this is more recent data with CRISPR, and it is interesting, as Dana asked, why it goes down here. We don't know that, what the reason for that is. Rewinding back to our oligo paper with ZFNs, this is work that Chandra Miller did in 2010. And so if you look at the total homology, so 50 on either side, all the way down to 15 on either side, she really saw it drop off, going from 20 to say 15. And so, it's if you look at how PCR works, oligos typically have to be about 18 base pairs or longer to work. So this is right in that range. So it looks like the chromosome needs, at least 20 or so to prime onto the oligo and copy it in. Okay, should the designs be sense or antisense? So this is another data set that Yiping and Xingzhou did. And so they tried 3 different loci, HER2, KRAS, and AVS1, and they're putting a restriction site right at the CRISPR cut site in each of these. And they looked at a positive strand and then an exact negative strand oligo of that. And so if you compare these numbers on either side, they're not greatly different. So when the mutation is right at the CRISPR cut site, there doesn't seem to be any preference for what strand the oligo is. So that's been our experience so far.
I'm sorry,
yes?
I didn't understand which strand is which relative to the guide RNA.
This is, I'll have to check on these guides. I'm not sure which, I don't have that data in here right now about, for each of these, which strand is targeted on the chromosome, and then which one of these, I'll have to go back and double check that to make sure that they controlled for that. So that they're, yeah, you're right. So that these numbers are relative, to make sure that this positive is relative to the guide RNA versus the chromosomal annotation. So.
They're not much different anyway.
Yeah, they didn't vary anyway, but I will go back and double check that. So. This is probably the biggest question and the most critical thing in terms of CRISPR design, because you only have a certain number of places you can design CRISPRs. And you want to get the mutation as close as you can to the cut site, because Dana was alluding to this the other day. Maria Jason has a great paper from 1998 that shows that as you put mutations near a SCI1 site, or as you move away, it goes down rapidly. And so here are 3 different loci where this Hindi3 site was moved progressively away. And you can see that once you get over 60 base pairs, you're really dropping significantly the amount of integration that you're getting. So our general recommendation is that the mutation, that whatever SNP or change you're making is within 60 base pairs of the cut site. So you may be able to go out to 90, you may be able to get away with it, but in general what we recommend to people is around 60 or so. And so this is maybe a little bit more complicated in terms of, it doesn't seem like when we do the sense or antisense here, there's no difference in this case in terms of whether you do one or the other. But I'm going to go back and show some things we've seen that are interesting with respect to moving this away. So, does it now, in this situation here where it's very far away from the cut site, you could ask the question again, does the strandedness matter as you move it away? And so everything we've seen to date kind of agrees with, I'll just call this a resection and priming model. It may be SDSA. Dana, maybe you can term it for me better. But this is sort of how, whenever we're thinking about making oligos, we think of this process happening on the chromosome. So you have a cut happen, and I'm only showing the right-hand side of the cut here. So during HDR, the chromosome gets resected, 5' end resected. So you wind up with a 3' overhang on the chromosome. If you throw an oligo in that's complementary, that 3' end can prime onto the oligo and start copying here. So I just put a green and red mark here to show the relative location of 2 different types of mutations. And so if you think about the chromosome extending from this point over, it's only going to copy in that particular mutation. It may miss this one and not copy it in. So where in the end you'll just have that. And so we've seen that. If we go back to the 2011 paper, we had an oligo that had several different mutations in it. So it had one that was on this side of the ZFN cut site. So we put these in here to disrupt, to keep the ZFN from recutting after integration of the oligo. And so if you see here, this is the number of sequence reads. We just did standard Sanger sequencing on a few clones. 29 of those reads, probably about 1/3 of them total, have all the mutations to the left here. And this oligo corresponds to the scenario here where it's copying in that direction. And so, but we also do see, another 1/3 of the time, that mutation does get incorporated when the oligo's that way. So I'd say the jury's still out on which, in terms of the strandedness for this. If you just wanted to pick one and go with it, you could think about it this way, in terms of designing the oligo when the mutation is far away from the cut site. So I would try to think with that mechanism in mind, because it seems to work for us. And then also, this is an independent publication from Ralph Kuhn's lab. So this is where he took some of the protocols we developed, and he tried to replicate the ssODN mutations in mouse embryos. So he injected ZFNs along with this oligo right here. So this ODN, that's the second to the last, has all these mutations. And he did something similar to what we did. He had ZFN blocking mutations on this side and on that side. And what you can see, if you look at PUP C7.3, you can see that everything to the right over here was not incorporated, but everything to the left, all those mutations made it into the PUP. So that agrees with some of this data that we saw as well. So, and when we think about it this way, all that makes sense. But as I said, there are some exceptions to that. Okay. So does anybody have any questions about this sort of oligo design? Because I think this is probably the most common application that people want to do. They want to model a SNP or make a codon change or something like that. And it's very rare that you're going to cut right at that, in this case, in the case that we were doing here, that's the conversion that our collaborator wanted, but we could only cut over here. It's very rare that you can get that break to happen right on top. So does anybody have any other questions about that? Yes?
Look at the impact of the number of variations that you incorporate in the oligo relative to the overall frequency?
Not in this scenario. In this one, we had 4, 5, 6 different mutations throughout that 120 base pair oligo. And we were able to get this BAMH1 conversion here at about 20% in K562 cells. We never did just keep ramping it up to see where it disappeared. Fu Chang, who did this work, in that paper, there's another novel thing he did where he showed he could design an oligo to delete a lot of DNA. So where you could make one of the homology arms homologous to a site that's really far away, and he found he could delete up to 50 or 100 kb off the chromosome just by designing the oligo a certain way. So that distant priming site, we were worried that could have off-target effects if it had other sites in the genome. He started mutating that, and I think when it got up to about 10% of the homology, it started to go down. So I don't know if that applies to this case here, but maybe if you got up to 10 or 20% of the homology, this would never prime again. So, yeah. Okay. So one other question we get a lot is, do the oligos need to be chemically modified? Because like I said earlier, there was a huge amount of literature trying to get oligos to work in human cells, and people used phosphorothioates quite a bit. So when we were publishing that paper, one of the reviewers asked us, why aren't you using chemical modifications? And all the oligos we used for that paper were not modified, so they were just straight oligos, standard synthesis. When we looked in the literature, we found this paper by Arts and Terrell, and I just pulled this part of the abstract out, and they found that oligos with the phosphorothioate linkages could actually cause double-strand breaks within the cells. And so you would get increased H2AX staining whenever you put those oligos with those modifications in. And then they would also arrest the cells so that they could never clone them. So we thought about trying to put these in and do them with ZFNs, but I used this paper to justify that we shouldn't have to explore that and never try that. And we haven't ever verified any of these results. But it's interesting to think about now because there's a lot of there's modified guide RNAs that are coming out now. I think there was a recent paper out of Matt Porteous's lab where they've put phosphorothioate linkages in the guide RNAs to protect them. And it makes me wonder if there's going to be some sort of adverse effects with this. And I've heard word of mouth that some people think this phosphorothioate, it's a sulfur group that replaces one of the phosphates in the nucleotide backbone. And they're thinking that it may react with cysteines, with the proteins when it goes into the cell, and just be gumming up protein activity within the cell somehow. So anyway, be aware of that. It'd be interesting if some people can try this, but just be aware that there may be some toxic effects of phosphorothioates at least. And so everything I've set up to now is standard oligosynthesis. So we've been doing a lot of library work with CRISPR now at Sigma, and that's largely enabled by technologies like custom array, where they can make 12,000 to 90,000 different oligos in a single tube. So you can order that for $1,000 to $1,200, something like that, and I have those within a week or so. So we use that to clone a lot of our guide RNA libraries. But there's a lab, I think Jay Schinder's lab recently showed that he made a library of plasmid donors. So you could now envision of trying to make oligo donor libraries. So instead of just making one oligo in a pool, make a ton of different oligos and then start doing screens. Like instead of doing different chromosomal sites, really mutagenize a certain area of a single gene to see what you could do. We haven't done any of that, but all the technology there is there to assess that. So, and you could use deep sequencing to tease all that apart in the end if you use lots of different oligos. So this is one question a lot of people have is, does the cell type support HR at practical frequencies? And we see this over and over at Sigma in terms of customer support. So we see people excited to make a SNP mutation. They design all the reagents and they apply them to their cell type and it doesn't work at all. And it just, I'd say 99% of the time it just winds up to be with that particular cell type not supporting HR very well. So we see that all the time. You saw a big variation here. This is from the oligo paper. And Actually, in this data set, if I had to predict ahead of time which one would have been the least, I wouldn't have thought it would be HCT116. So when we did this with plasmids, HCT116 is known to do HR quite well on its own, but it doesn't do oligos very well for some reason. With plasmids, MCF7s are horrible. We can hardly detect any HR in MCF7 with a plasmid-based mutagenesis as a donor, but with oligos, it seems to work at 20% levels. So if you're having trouble getting HR to work in your cell type, you might try plasmids and oligos to see if one of them if there's a preference for one or the other. Yeah. Okay. So last thing I'll talk about here is ploidy. So I'm kind of a visual person. I've never done cytogenetics before we had ZFNs in, but I really like looking at karyotyping. So can anyone tell me what's wrong with this particular karyotype? Anyone an expert out there? Anyone know what's wrong with this? So those are actually socks. So this is at the Guy's Hospital in London. So they have a big piece of art up where they've taken a karyotype. They just took some striped socks and stuck it together. So anyway, so they do look like socks when you look at these sky karyotypes. They look like this. This is what normal human cells look like. You should think about this when you're doing gene targeting, how many copies your cell has. So if you're starting out in the lab and your PI wants you to work with KBM7 cells, they're haploid, great, that's going to be easy. But if your advisor told you to knock something out in this cell type, which is C70, and there's almost like 6 or 7 copies of every chromosome in that cell, I mean, it would be really bad. So I think that's an extreme example. Here's, HeLa is in more common use, but still you can see examples where there may be 5 copies or so. And then on top of that, it's all rearranged. So there are parts of one chromosome latched onto another chromosome, making it difficult to know how many copies you actually have in some cases. So when you do knockouts, you can kind of get around this by screening for activity. If you have a good assay for the protein you're trying to knock out, you can hit the cells, screen clones, and see if the activity is gone, and then genotype it. But when you're doing HDR and SNP conversions and things like that, you'll want to make sure that you have conversions in all the alleles. And if you have a cell type like this, you're going to have an uphill battle. So it's something just to be aware of. So to give you some resources to look at that, this is the SCI database at NCBI. So if you just do, SCI database, NCBI, just Google that, you'll find it. There's a lot of different cell types that they have logged into there. And so they have all these laid out. And if you mouse over this, like if you put your cursor on it, it'll tell you if you're on the P or Q arm and like what section you're in. And then you can use, if you see something, you can find that out from a genome browser where your target site is, and you can go and find and estimate how many copies you have. So that's a low resolution way to look at it at NCBI. If you want higher resolution, the Wellcome Trust at Sanger has a great site called Cosmic. So if you want to look at K562 cells, you can type that in. And then they have much higher resolution maps of chromosomes where you can look at copy number. And so if you're wondering about targeting a certain region in a cell, you can go look at this and get an idea of what you're in for to look at. So that's all I've got for right now. There's a lot of other stuff I can talk about with what Sigma does. We've been working on genome editing for about 10 years or so. And there's no way I can summarize it quickly. So I thought I would try something my 11-year-old taught me. So he had to do these, in school, he had to do these things called Wordles all the time. So he would get a spelling list every week, and he would have to put all his words that he learned how to spell into this Wordle, and it makes a graphic for that. So all these words here, we do all these things at Sigma. So either in R&D, either in an R&D mode or in just a regular product development mode. We still work with ZFNs. Lentivirus is a big thing for us with shRNAs. We do a lot of custom work for cell engineering, for CRISPRs and design, lots of different things. So with that, I'll stop right there. Few people I want to thank. Primarily, in terms of getting started with ZFNs and Targetron and CRISPR, is Xiaoxia Sui. So she came from Alan Lambowitz's lab, and she she really got Targetron established at Sigma and also got ZFNs established. I think she's a VP of R&D now at Horizon Biotechnology. Also like to thank Fucheng Chen. He did a lot of the oligo work, really did a lot of hard lab work, a lot of creative, intelligent creative work in terms of getting everything designed and working out rules. Also Chandra Miller with a lot of the oligo work. She was a big help. She is now the director of the Genome Editing Center at Washington University. So they have an IPS and a Genome Editing Center. So she came out of Matt Porteus's lab. She has a lot of experience there, working there currently. And so this is the facility where we work at Sigma. So this is mostly a biotech building, does molecular biology research, where our lab's located. So this is the current group we have doing a lot of the CRISPR research right now. This is Fucheng. We also have Xingzhou Ji. He's working on a lot of activator work at the moment. And I'd also like to just take a moment to plug our science education outreach. So Sigma has something called Science Partners. And what it does is it allows employees and a lot of the scientific groups to go out into the community, to high schools, and run scientific experiments. So in this case, this is a chemiluminescent protocol that they took out to one of the local high schools. And I can show these pictures because they wound up in the paper, so they wound up in a local newspaper. So I was really proud of them for that. Ashley Fisher in the group really loves this, doing this. I think she's trained about 1,500 different students now through this program. And so Sigma is very supportive of that. They also have a great intern program. So we had Rochelle Wong last summer from UCLA. She did a lot of work for pre-work for some of the pools that we were developing with HPRT1 gRNA. And so we really enjoy having interns. So if you have any students in any of your labs or any of you guys are interested in internships in industry, just let us know. So I'll stop there. Thanks a lot.